Thanks very much, and thank you, all of you that are here, and for your continued attention for the last few minutes as we complete today's sessions. What I'm going to try and do over the next few minutes is give you an update and an overview of some of the exciting work that we're doing at Athersys, both internally and also in collaboration with some outside groups that we work with. I will be making some forward-looking statements that are covered under Safe Harbor, so I refer you to our SEC filings and related information that's available. You can go to our website or to the SEC website as well. As someone that's been involved in the meeting on the Mesa since the very early days and since the early days of the formation of the Alliance for Regenerative Medicine, I have to say it's incredibly gratifying to see the tremendous progress that has been made across and throughout the field. Here at the conference this week, we have over 1,200 attendees that are representing companies large and small on a global basis that are displaying some of the exciting progress that's being made in the field of regenerative medicine. I think this really, in many ways, represents a very important demonstration of the fact that the ecosystem is working together in a lot of collaborative different ways and is in the process of transforming medicine as we know it. At Athersys, we're very proud as being recognized among the leaders of the companies that have been involved in this space for a long time. During the presentation today, I'm going to focus on some of our key programs. Our emphasis is really on using our proprietary off-the-shelf cell therapy platform that we refer to as MultiStem for indications in the critical care spectrum. I'm going to give several specific examples of that during the course of the presentation today. Our most advanced program is in phase 3, and this is covered under Fast Track designation. It's also received RMAT designation. And so I'm going to spend a portion of the presentation actually talking about that exciting trial, which is to treat patients that have suffered very serious and debilitating ischemic strokes. In addition to that, I'll spend a few minutes talking about another exciting program where we recently announced some data in treating patients that are suffering from acute respiratory distress syndrome. This has actually led to recently our third Fast Track designation. In fact, I think we're one of the only cell therapy companies that has received 3 Fast Track designations from the FDA and similar designations from other regulators around the world. We have a very important partnership which was discussed yesterday in Helios's presentation. I'll touch on that a little bit later during the course of the presentation today, where we have 2 active clinical programs, or we are supporting 2 of their important clinical programs, both in stroke as well as in acute respiratory distress. Behind that, of course, and again, I won't have time to cover all this today, we have a very robust clinical and preclinical pipeline that I refer you to our website for more information. We also have a very solid financial position. Now, as I mentioned, we're heavily focused on the critical care side of things, and we do this because many examples or many publications have shown that this is where our technology has particular relevance, but we also do it for a more pragmatic reason. The critical care spectrum and indications that we're focused on, we know that we can use our technology and actually get to efficient answers about whether or not administration of MultiStem is helping patients after they've suffered a devastating event, whether it be a stroke or some of the other things that I'm going to be talking about today. And so we think, in effect, this gives us an expedited path to getting answers and allows us to put us in a position to actually achieve results that really matter and that can help us deliver on the promises to our shareholders. So MultiStem cell therapy is something that we've been working with now for quite a number of years. This technology is based on the proprietary research that was done a few years ago. The important aspects of this are that the cells that we work with have some unique and distinctive characteristics. The cells that we work with are obtained from healthy consenting donors, typically in their mid-20s. When we isolate these cells, we and many others have shown that these cells have robust growth properties, so we can actually grow them up in enormous quantities. And in fact, as we published and described many times, the amount of material that we can get from a single donor bank actually allows us to produce the equivalent of millions of clinical doses of material, or even beyond that. Importantly, we've also shown clinically and through years and years of preclinical work, we can administer these cells just like type O blood. We do not have to tissue match or immunosuppress the patients that we are administering MultiStem to. So those 2 things in combination, the scalability aspect of this and the fact that we can administer it as a true off-the-shelf type of cell product, are very, very important. We have a very robust IP estate on this technology. We have now over 300 issued patents that cover our intellectual property estate. We also know, and I'll describe this further in the presentation, that these cells promote healing and tissue repair through multiple distinct mechanisms. So they're unlike traditional pharmaceuticals, unlike traditional biologics that do one very specific thing. These are dynamic living entities that can actually regulate the healing and tissue repair process in multiple different ways. We also know that the product has a long shelf life. In fact, we've got over 7 years of stability data that shows that when our cells are properly cryogenically maintained, they are stable for a very long period of time. We've worked for the past few years to really simplify the product preparation and administration to really boil it down to a couple of key steps that are shown here. And I'm going to elaborate on this in just a minute, but the point is that by doing this in a true off-the-shelf type of format, we think that this helps unlock the potential of this form of cell therapy and regenerative medicine, making it easy to use. It can be accessible right through the hospital pharmacy. The 3 steps in product preparation are remove the vial from the freezer, thaw the contents, which are in frozen form, and then transfer it using a sterile syringe into an IV bag of saline. Most of the indications that we administer MultiStem for, we administer the product intravenously. We wanted to make it so simple that virtually anybody could do it with nominal training and also kind of non-threatening and easy to implement. And in fact, we've succeeded on that, and that was a very important step. But we haven't rested on our laurels, so to speak. For the past couple of years, we've been developing additional technology that we think really simplifies the ultimate delivery of this technology to hospitals around the world. So a couple of years ago, we really began to focus in earnest on developing a technology referred to as SIFU, which is a proprietary cryogenic storage system that does not use liquid nitrogen. And as a consequence of that, this is the type of technology that can be placed in virtually any hospital pharmacy or other parts of the hospital at any clinical institution where we want to utilize or place the technology anywhere around the world. Importantly, this technology platform enables us to provide secure access of the and protection of the inventory. The inventory is protected at all time in a safe, stable, secure environment. It also gives us real-time informatics access, so we can always see the product, if you will, even remotely, which is very, very important to make sure that you're monitoring for product integrity and being able to see who's getting access to the technology. Importantly, it also allows us to comply with track and trace in a very robust way from end to end, and this is very, very important. And it also simplifies the product preparation down to a single step after the order entry is completed, which again just is very simple and straightforward. And as you can see here from the diagram, or from the image of the SIFU here, there's a dispensing port right here which actually dispenses the thawed and prepped cell product directly to the pharmacist or the whoever else is actually accessing this, so that they can then transfer it to a bag of saline in a single step and take it to the patient. It doesn't require any special facilities, and it doesn't require extensive training. So again, we've designed this so that it is as simple as possible to use. We've also spent a lot of time over the past few years advancing and refining our product development technology and approach, and in fact, we've talked a lot about this publicly in the past. We are committed to working in larger scale bioreactor platforms. That's something that we've been working on for a number of years, and in fact are actively transferring our bioreactor platform capabilities to the GMP contract manufacturing environment. And we think that ultimately this will help us unleash the full potential of this technology. One of the things that we know about MultiStem is that these cells do not permanently engraft when we administer them to patients, but they do regulate the healing process in multiple very important ways. They decrease the inflammatory process that occurs after a significant event, whether it be a stroke or some of the other things that I'm going to be talking about today. We know that these cells also upregulate key reparative mechanisms and pathways. They stimulate other cell types that are important in the healing process. So things like regulatory T cells or M2 macrophages or other pathways or other dimensions of the immunomodulatory component that help ultimately the body recover from something really bad that's just happened. We also know that these cells stimulate neurotrophic or neurogenic responses when there is a neurological injury, for example, and they do other things that actually help the body and the patient recover in some very important ways. Because of the evidence that was accumulating over quite a few years of research, this ultimately led to advance our clinical program in ischemic stroke into clinical development, and I'm going to walk you through that in just a minute. I think most of you here already recognize that stroke is the leading cause of serious disability in most countries in the world today. It represents a massive problem on our healthcare systems throughout the world. And it's something that really suffers from very limited options in terms of treating stroke patients. We think we can change this. And one of the reasons why we think we can change it is because we have clinical data, which I'll summarize for you in just a minute, that shows that we can treat stroke patients up to 36 hours after a significant ischemic stroke event has occurred. This is a window that's well beyond the historical norms for standard of care, and something that we think will allow us to reach many stroke patients that might otherwise have no treatment options, and essentially they're relying on palliative care or rehabilitative therapy, and frankly that doesn't help a substantial percentage of the patients that desperately need help. We also know how the cells work. We know, based on work that we and other independent labs have done over more than a decade, that after we've suffered an acute neurological injury like a stroke, but it also involves other events that can occur, that actually causes a subsequent activation of the peripheral immune system. This work that's shown here was not done by us. It was actually done by an outside independent lab a little over a decade ago, but it was consistent with the results that we had already generated and work that we were already doing that illustrated the importance of the peripheral immune system. And without walking you through the entire set of images here, it's important to note that when the peripheral immune system actually mobilizes following a stroke, it creates a massive amount of hyperinflammatory activity in the region of the stroke or ischemic event in the brain, and this can be devastating. What this work shows is that if you remove the spleen before that actually happens, you neutralize the substantial amount of damage that is actually being done. You still see damage due to the ischemic event, but it's a lot less because of that hyperinflammatory cascade is no longer in effect. But surgically removing the spleen is not really the best way to treat stroke patients because it would leave them immunocompromised for the rest of their life. But based on the work that we and others have done, we've shown that if we administer MultiStem in a clinically relevant window, which is again up to 36 hours post-stroke, we've shown that these cells can actually go directly to the spleen, they downregulate that hyperinflammatory cascade and essentially neutralize it before it begins to get out of control, and at the same time stimulate key reparative mechanisms that help the body and the patient recover over time. We've also published on this. I refer you to a recent example from 2017 that illustrates the depth of our knowledge. We have other publications that talk about what we know about how MultiStem relates to this. And in fact, we're one of the most published companies, I believe. Even if you go to our website, you see that we have dozens and dozens and dozens of publications in leading scientific journals working with outside independent collaborators. This illustrates a very important cultural point at Athersys, which we believe in publishing our results, and we believe in encouraging the people we collaborate with to publish the results as well. Just briefly, I'm going to touch on the results from our Masters 2 study. I think one of the things that's important for people to understand about the stroke area is there are two well-accepted regulatory endpoints for stroke clinical trials, one being excellent outcome, which essentially is the proportion of patients that achieve full recovery, and then something else called the MRS shift analysis. I'm only going to talk about excellent outcome today. This was a double-blind randomized placebo-controlled trial that was conducted at 33 leading stroke centers across the US and the UK. And this data, which we published, showed quite clearly that when we treated patients in this study, whether it was all subjects enrolled in the trial, some of which were actually treated beyond that 36-hour window, that by the time we completed the follow-up assessment at 1 year, we saw very compelling evidence at 90 days, but we saw even more compelling evidence at 1 year. And when we looked specifically at patients that were treated within 36 hours or less, we saw that there was dramatic evidence that these patients were recovering at a much better rate and to a far greater degree than patients that were being treated under standard of care. And it was highly statistically significant. This is just a slightly different way to look at the data, and what I would encourage you to do is actually look at the image on the right. This shows the patients that were treated within that 36-hour window, and you can see there's a very different trajectory, and that's because we know that we're influencing the environment in the brain that helps these patients recover and heal more effectively over time. So again, just another piece of significant evidence. We also prospectively designed biomarker analysis as part of the trial. This data was entirely consistent with and showed that our therapeutic hypothesis was in fact being validated by this evidence. It showed that we were reducing the level of activated immune cells and also the level of inflammatory cytokines that are key culprits in the inflammatory pathway. And importantly, we also showed that among more severely impacted or disabled stroke patients in particular, we were showing that there was a clear difference between the incidence of life-threatening and severe adverse events, which is something we predicted when we set this trial up. And in fact, this data, which was a post-hoc analysis, showed pretty clearly that that was in fact happening. All of this formed the basis for a very important partnership that we announced back in 2016 with Helios. They gave a great presentation yesterday, which kind of detailed some of the things that we're doing together or that we're supporting their efforts to do. And a lot of this is being done under the new accelerated regenerative medicine regulatory framework, which was implemented in Japan going back several years ago. Last year, we announced a meaningful expansion of this partnership with Helios, which allowed us together to move forward in some other areas that are pretty exciting, including the ongoing clinical trial that they're conducting in acute respiratory distress syndrome. And in parallel with that, we're running our own phase 3 clinical trial, pivotal study that's being conducted under an SPA, and as I mentioned earlier, with Fast Track and RMAT designations. And importantly, we're applying all of the key lessons and learnings from the last study in this trial. So in the last couple of minutes, I just want to focus on some of the other critical care programs that we're focused on, in particular focusing on ARDS. ARDS is a severely disabling condition where patients, their pulmonary capacity and function has become so compromised, they've got to be placed on a ventilator and they're being cared for in the intensive care unit. It affects a lot of patients in the United States and in Europe, and also, as Helios described yesterday, a meaningful number of patients in Japan. It also represents a major area of unmet medical need, because really the only thing you can do for these patients is put them on a ventilator and hope for the best. And hopefully these patients will survive long enough where their pulmonary function begins to rebound. You can wean them off the ventilator. They typically remain in the ICU for a period of time after that, and then typically you can move them to less intensive hospitalization. We conducted a study, and the principal investigator for the trial, one of the foremost experts in this field, presented the results at the American Thoracic Society meeting just several months ago. What this data showed, and again, this was a double-blind, randomized, placebo-controlled trial, was that when we administered a single dose of MultiStem to patients that were suffering from ARDS, particularly patients, as shown here on the bottom, that were suffering from more severe pulmonary function or more severely compromised pulmonary function, that it had a dramatic effect on the trajectory of these patients. We were showing a significant increase, or a substantial decrease in mortality. We also saw meaningful improvements in ventilator-free days, in ICU-free days, and so across the board we were seeing encouraging evidence, and this was supported by the biomarker evidence, which was also described during this presentation at ATS. Importantly, from a Helios perspective, this put them in a very good position to then move forward with their study, which they are now conducting in Japan, which they refer to as the One Bridge study. Now, our study focused on multiple different types of ARDS. In Japan, they're focused specifically on pneumonia-induced ARDS, so they asked us to do an analysis on those types of patients, and importantly, the data showed there was a very strong response among patients that had been diagnosed with pneumonia-induced ARDS, as shown here. We basically doubled the number of ventilator-free days and the number of ICU, more than doubled the number of ICU-free days for these patients within that initial 28-day assessment period, and had a substantial impact on reducing mortality as well. Another program, and the last one that I'll have time to mention today, is what we're doing in trauma. This is a DoD-funded program. We are in the late stages and getting close to finalizing the regulatory preparation for this study. This will be a double-blind, randomized, placebo-controlled trial, again with support from the Department of Defense through their MTech funding arm, working in collaboration with one of the foremost trauma centers here in the United States, if not in the world. Interestingly, our data and the data generated by work that, with collaborators shows that the same type of hyperinflammatory response that occurs following a stroke or that occurs following other types of events is directly relevant to what happens to patients that have suffered a trauma. And we believe, again, once that happens, these patients begin to spiral down. They experience organ failure, multi-organ failure, or other things. We believe that we can change that picture. Trauma is a major area of unmet medical need. Most people don't realize that it's the leading cause of death among people age 45 and under, and it's also a leading cause of serious disability. So again, like the other critical care indications that we're focused on, we think we can have a big impact here. We made a lot of progress over the past couple of years in particular that we're very proud of. I'm not going to walk through all of this, but again, I refer you to our website. One thing that I would, just in closing, like to point out is that one of the collaborators we've been working with in the UK, Dr. Emily Thompson, was just recently awarded the Da Vinci Prize by the European Society of Organ Transplantation for work that we've been doing and that she's been spearheading in the use of MultiStem in the transplant space, particularly focused on renal function. We believe that this award was well-deserved and is a great acknowledgement of the pioneering work that she's been doing and that we've been supporting with her, and we're very excited about that. So with that, I'd like to close, and I'd like to thank you all for your time and attention, and I hope you have a good night and enjoy the rest of the conference.